you see your future in a scientific career? Is it the class you most enjoy in school? The Junior Science and Humanities Symposium is a marvelous way to pursue your interests.
Symposium has been in existence for 45 years, and Northern California, Western Nevada started at the same time as an original region.
As science and technology have changed, originally our symposium, the kids had to use, everything had to be done by themselves. Now they push a button and you get the results.
It's an opportunity for high school youngsters who have done research, original research, to present what they have done in the format of a scientific meeting.
Symposium has actually two aspects. It is a scholarship competition.
For a lot of their peers and their teachers and so forth to be there as observers and to interact with them.
And interestingly, the top student in each of the 6 categories at National Symposium then are selected to go on to the London International Youth Science Forum to represent the United States.
Most of the participants are here as observers to find out what's going on, to interact, to maybe get ideas to do research.
We have over 100 students and more than 50 teachers who come to experience the university campus, to hear the presentations, to interact with the university in many ways. They'll have roundtable discussions with graduate students in the sciences. They'll have visits to research and development and educational activities on campus. They'll hear nationally known speakers.
The symposium offers another real important way for me to stay involved, to work with kids, listen to kids, talk with kids, and support a great program, a great opportunity for kids.
In accordance with my hypothesis, my antibody was specific to Marburg antigens only. It is also important to note that it did not cross-react with Ebola antigens. This is important because a monoclonal antibody fighting a virus should be specific to the antigen you are treating it with.
My hypothesis consisted of the fact that the polymer would be formed and it would be a gel. And I also believe that the polymer would be equally effective in removing both acidic and basic contaminants, because the cavity itself didn't seem like it would be especially picky on which type of contaminant it would attract.
Usually it doesn't follow the regulation. There's always a residual accumulation of fluoride. So let's say if you had a water level of a lake and it drops, the concentration of the fluoride in the water will increase.
And I would like to develop a formula to predict the urban heat island effect for small cities so that city planners could use it to mitigate the urban heat island effect.
From about 40 participants, we had to narrow it down to 16, and all of the 40 were quality projects, so it makes a daunting project for the judges.
At each of the regional screening sites, sub-regional screening sites, there's a panel of judges from Cal State Fresno, from University of Nevada, Reno, from UC Berkeley, and from UC Davis, made up of faculty, and they hear the oral presentations and they rate the students on their involvement with the project, with their ability to communicate the project in a PowerPoint oral presentation as being ready to present, or almost ready to present, or maybe not quite ready this year.
I don't know of any other stage like this that allows students to take the experience that they have, the interest that they have, the work that they've done, whether it's, I just heard a student that did her whole project at home. Some students work in the summer seminars at a local university or college. Some have science projects at their school that they work on, and here's an opportunity for them to take that work, that research, and showcase it with other kids with similar competencies, capabilities, and interests.
We've been trying to figure out what exactly a certain protein, which is called CLC3, does in the cell. And this is important because it controls certain electrical impulses, especially in the heart and the vascular system. It facilitates the transport of chloride channels from outside of the cell into the cell. These chloride ions are essential for the heart so that it can keep beating. So it's a very big protein, and we think that it may contribute to certain heart problems and other diseases regarding the cardiovascular system.
We're trying to teach them the basics. This is how a research scientific idea emerges, how it develops into a hypothesis, how an experimental design is set up to address this hypothesis, how we interpret results, and how we come up with the next hypothesis, which is now based on something that we have established already experimentally.
Dr. Jan Believe and his lab technician, Jan Dingdai, have been very helpful in helping in my learning experience. They've both taught me amazing amounts of information, and also they've also been kind enough to just let me sit in here and watch when I'm really more of a liability in the lab than anything else, and seeing how I didn't have any idea of what goes on here before I came in here.
In the beginning, you see these young students come in the lab knowing almost nothing, with no background, and then you see these sparks showing in their eyes until at the end of the training they get out of the lab knowing a lot, being able to give great ideas about the research, and many times being able to carry out the experiments themselves. And this is really a very rewarding experiment.
Right now, I think it's more academic, just trying to figure out what's going on in the body and why. But I think that this research, once it's progressed to a certain level, will be essential in curing certain heart-related diseases and also stopping heart attacks in the future.
Well, I think we all want to make a difference. You know, we all have our passion to follow, and you know, this is one way to do that. It's a great tradition with the symposium, and it's a real honor, you know, to be involved in continuing that tradition. So if there's a student out there that's interested in following their interest in science or math or humanities, follow it. Come to the symposium. Tell us about it.
If you are interested in the Junior Science and Humanities Symposium, there are two ways in which you can participate. The first is by talking with your science teacher, asking to attend the symposium as an observer. The second way is in becoming a presenter. Your science teachers, GATE, or GT facilitators can help you find a mentor working in the field of science to facilitate your research. Participating in the Junior Science and Humanities Symposium could be the open door to your future career in science.